Yeah, it should be out now. So I really want to thank the organizers for the invitation. It's really nice to drive up here without traffic this morning. So I'm just down the South Bay. I'm a junior faculty at the neurosurgery department. So my lab is really interested in study fine movement control. I want to continue the topic on basal ganglia circuits. So as you can see, for this baseball player, in order to hit a perfect home run, so you have to have an instantaneous integration of motor and sensory information, right? So you have to have your target, you have to constantly judge the speed and direction of your balls, so your brain is doing all kinds of computation. So my lab obviously cannot study the baseball players directly. We use mice as a model organism. For example, we can train the mice to perform a full reaching task and study the fine motion. But today I'm not going to talk about behavior. I'm going to focus on the neurons that are important for encoding these sequence of motions. So for example, Josh has nicely alluded to the circuits that is responsible for fine movement control. I'm not going to go detail into the circuit architect. I just want to mention that the basal ganglia, which is a group of interconnected nuclei, is very important for integrating these convergent synaptic inputs. And not only important for movement control, but also for a variety of other functions here. And the whole circuit is heavily modulated by dopamine signals, like what Josh mentioned. And dysfunction of the dopamine signal can lead to movement disorders and also various forms of addiction behaviors. So we really want to understand the underlying cellular and the neural circuit mechanism in these neural circuits. So how do we do this, right? Many of us actually think about the brain like computer circuits. So in order to understand the brain, we need to understand the circuit. The basic fundamental question is try to understand how these neurons integrate the inputs. So to do this, we imagine, for example, the central neurons in certain brain region as a computational elements. So as we give inputs, and then we can spit it out output. So in my talk, I'm going to focus on the spiny projection neurons in the striatum, which is the principal projection neurons in the striatum. So how do we study this? There are many, many ways, right? So for example, here I show example the work done by Hongwei Dong's group. So you can do large-scale circuit tracing. For example, in this case, use wire space tracing. Look at the fine projections from various parts of the cortex into different parts of the striatum, right? So this way you can build a large-scale whole animal scale of projection levels. This is anatomical data. Obviously, this is not enough to understand the function. The other way is to read out the output, similar to what Josh does, for example, recording the activity of the neurons. In this case, I show an example done by my colleague Mark Schnitzer using the miniscope and then simultaneously record hundreds of neurons' activity in behaving animal. So my lab actually does neither of this. We really try to understand the cellular and the subcellular function that is bridging these findings. So come back to the spiny projection neurons. In order to understand the computation, we really need to know the inputs and then know the outputs, right? So what kind of inputs do these neurons receive? They receive a variety of excitatory inputs and modulatory inputs. For example, motor cortex, as you can imagine, we need motor commands generated in the motor cortex. At the same time, we need the sensory feedback to adjust our motion. These are primarily coming from the S1 somatosensory cortex. And also, we also need to respond to salient stimulation and the attentional shift signals. These are mediated by thalamic inputs, primarily parafascicular and central medium nuclei. And all of these aspects are heavily modulated by dopamine inputs, like what Josh mentioned. So how do we understand this computation and how these inputs are computed in the single neuron level, generated the action potential output? This is what I'm going to focus on today. So I'm going to try to answer three fundamental questions. Actually, many of us have been trying to tackle for many, many years. First is, many of these convergent excitatory synapses coming from, for example, M1, S1, and thalamus, they are all glutamatergic. Do they actually form some kind of organized synaptic inputs onto the dendrites of median spine neurons. And if they do, how these convergent synaptic inputs are computed on these dendrites and translate into spiking outputs. And lastly, if I have time, I'll mention, these spiking, how are they fine-tuned by the inhibitory network within the striatum. And of course, these inhibitor inputs are also driven by these excitations. So how do we do this? Actually, I'm an experimentalist. In our lab, we primarily use experiments to measure the outputs and control the inputs. And then recently, we started exploring using computational methods to guide our experiments. So how do we do experiments? We use combined electrophysiology and two-photon imaging. We can perform whole cell patch clamp recordings in the targeted neurons, and then record the synaptic activities or spiking activities in response to either current injections or synaptic stimulations. At the same time, we can load the cell with dye to guide us to study the morphology or targeted stimulation. And simultaneously, we can look at calcium signal in these neurons in very fine structures such as dendrites or spines. So in order to understand the computation, first we really need to precise control the inputs using genetic methods. So thanks to invention of optogenetics, we can express channel rhodopsin in very specific areas in the cortex or thalamus, and then stimulate these inputs and look at the output. Right? For example, we use EMX1 Cre, in which Cre recombinant is expressed in all cortical neurons, either by crossing with the AI32 mice, where channel rhodopsin can be expressed in the Cre-dependent manner, or we can inject AAV-mediated Cre-dependent expression of channel rhodopsin. In this way, we can control the channel rhodopsin either in all cortical areas throughout the cortex, or in the defined region. For example, in this case, it's S1. And this is how we express channel rhodopsin in motor cortex. We can use a different Cre line, bigly T2, for example, in this case, to express channel rhodopsin in thalamus. And these are target area. What if we want to do sparse labeling? For example, we can use Thy1 channel rhodopsin YFP line. In this case, random group of layer 5 parameter neurons in the cortex express channel rhodopsin. So provide us additional control, we can stimulate a subselective group of neurons in the cortex, or a target group of neurons in the whole entire region. So all these manipulations can nicely express channel rhodopsin not only in the cell body, but also in the axonal terminals, so which allow us to record and stimulate in the brain slice. For example, in this case, we can record the postsynaptic neuron, load the cell with the morphology dye and calcium dye, and locally we can activate these channel rhodopsin expressing axons, and then we can observe a calcium transient in the target area. In this case, we can define a functional connection between the channel rhodopsin expressing axons and the postsynaptic spines. So this is how the real expansion works. You can pay attention to the blue dot here. This is when the channel rhodopsin stimulation is turned on, and then you can see calcium transient very defined in this spine. So we can do this way, and repeatedly again and again through a dendritic arbor. And we do this multiple times because synaptic transmission is probabilistic. We want to capture the most functional connections. So this is how data looks like. If we go through the dendritic tree, in this case, 2 major dendrites in this one spiny projection neurons. In this case, the channel rhodopsin expressed in the S1 corticostriatal projection axons. All these red spines are marked along the dendrites, indicate the functional synapses coming from the S1 corticostriatal projections. Okay, so if you look at these distribution, we did this in many spines and many neurons. A few obvious conclusions can immediately made. First is the inputs coming from the S1 are not segregated into different dendrites. Any dendrite we look, we always have inputs along the dendritic tree in the region where the neurons receive S1 inputs. And the other simple conclusion we can make is that the S1 inputs do not have distal or proximal preparations. For example, famous CA1 pyramidal neurons in the hippocampus receive different inputs in the distal tufts versus in the middle or versus the basal dendrites. This is not the case for striatal projection neurons. So what are the organization, right? So the first thing you can notice that we do see some kind of clustered inputs that are congregating the subregion in the dendrites. Is this true? We performed different levels of cluster analysis. One of the example is the nearest neighbor test. In the middle is more random distributed dots. On the left is more dispersed, and on the right is more clustered. By calculating the nearest distance with its neighbors, we can get a reliable measure of t-scores and p-values, where the on the right is indicating is perfectly dispersed inputs, on the left is perfectly clustered. So our data gives a very nice indication of clustered inputs when the channel rhodopsins are expressed in either somatostatin cortex or the motor cortex. And this is not true when we randomly express channel rhodopsins throughout the cortical regions. In this case, we used Thy1 channel rhodopsin, as I mentioned before. The inputs are randomly distributed, and we have a p-value way greater than 0.05. And this is also not the case for thalamic inputs. In this case, we express channel rhodopsin using VGRU-T2 Cre mice, and you can see also the inputs are perfectly randomly distributed throughout the dendritic tree. So what this tells us? This tells us actually cortical inputs, specifically from S1 or M1, are clustered along the dendritic tree. But how clustered is the cluster? In order to estimate how big the cluster is, we use the Ripley K function to do the analysis. So what is this analysis? You can imagine, yes?
Jim, I'm a little confused because you're showing a 2D picture here on your
Yes.
cluster analysis, but of course the synapses are constrained to lie along the dendrite.
Yes.
So
We use a modified 1D version of it. It's just 2D version is easier to explain. That's why I used for the background. Yeah. So again, for the Ripley K function, we also use a modified 1D version of it. So what is the Ripley K function? So you can imagine you're walking into a botanic garden, right? All the rose bushes are together, all the conifers are together in the Japanese garden. So this is the way to estimate how clustered of these same plants in the same region, right? So by measuring the distance and between other objects within the radius, for example, in this case, we can generate a random distribution. If the true distribution is above this random distribution, it suggests a significant cluster. For example, the rose bushes in the botanic garden. Or below this line is significantly dispersed. For example, the trees along the main street in most towns, right? So we use 1D, modified 1D version of analysis and look at the cortical inputs. In this case, you can sort of see already, right here.
Is KFR just density?
What?
Is KFR just the density?
What?
Is KFR, this Ripley's K function, is that just the density as a function of R?
It's more than just density. I think it takes more than a few minutes to explain. We can talk afterwards. Yeah.
Other questions?
Yes.
You know that it's different axons for your cluster?
Yes.
Okay.
Yes, we do. Yes. So these clusters from cortex, yes, indeed, is a significant beyond the random distribution. And if you perform the same analysis, in this case using Thy1 channel rhodopsin mice, you can see that the true distribution falls in the 95% confidence interval of the random distribution. So we can do this in many, many dendrites, in many, many cells, and estimate how big the cluster is. So by estimating the region that is beyond the random distribution, which gives a measure about cluster length about 30 microns. Within this 30 micron cluster, we get about 10 spines that are clustered from specific region of inputs. Why this is important? Because we think it's actually an important way to encode the different ensembles doing specific behavior. For example, if the ensemble of activity and their inputs are converged onto the same dendrite, can way enhance the computation of these striatal projection neurons. But if you sparsely label cortical neurons throughout, or thalamus neuron, and then they randomly distribute, they can mediate different level of computation. So this is the estimation, right? How do we measure the computation? So let's move on to the second part. So look at the distribution, right? The first thing we want to do is, can we actually co-activate these synaptic inputs at the same time? So the way to do it is to use two-photon caging of caged-glutamate. This is a compound of caged-glutamate. The red circle is an active glutamate compound. Upon absorbing photon at the right wavelength, this compound can be cleaved and release glutamate. Because two-photon excitation volume is extremely small, we can precisely control where we activate these synaptic inputs. For example, here I show this targeted spine. If we simply move the uncaging spot 1 micron away, we can lose the synaptic response. So this is a very precise way to target the individual spines along the dendritic arbor. So we can do this, for example, on this specific stretch of dendrite. We can target these spines one by one and in the random way. If the dendrite is computing in a linear fashion, we can calculate the arithmetic sum. If the dendrites are calculating these inputs in a nonlinear way, we can compare the measured response with the arithmetic sum, which generates this curve. You probably already can guess the dendrites actually computing these excitatory inputs in the superlinear way. In this specific case, the ninth spine is already significantly deviated from the standard unity line. So this is not surprising. This phenomenon has been shown in many types of other neurons. Co-activation of clustered synaptic inputs can enhance the dendritic integration. This is obviously, in addition to a simple dendritic computation, right? So if you compute the inputs in the linear way, you can put a threshold and generate action potential yes or no fashion. And then in the nonlinear way, up to a certain threshold, the activation of cluster inputs can generate this big plateau potential in the dendrites. And actually, a paper from Bartlett-Smelt's group more than a decade ago has nicely shown in theory the dendritic nonlinear integration can enhance the computation capacity in the single neuron levels. So how does this mean for the striatal projection neurons? Well, we need to look at the action potential outputs, right? But we can only stimulate a few dendritic spines. Obviously, this is not sufficient. So we really want to test the idea that if the dendritic nonlinearity happens, and then we can activate other synaptic inputs at the same time, does this facilitate action potential generation? To do this, we collaborate with genetic algorithm group at the Karolinska Institute, and Kai Du, her student, generated a detailed model for spiny projection neurons using the realistic morphology of striatal spiny projection neurons. And this model has been recently published and being available on the ModelDB. So in this neuron, we can insert all the known ion channels and synaptic conductances using the known distribution published in the previous papers. So this model can very faithfully recapitulate the excitability of the neuron. For example, in response to current injection or spontaneous synaptic inputs or inhibitory and excitatory and inhibitory inputs. And how about non-linear events? As I mentioned, if we do two-photon glutamate caging in the stretch of dendrite, we can evoke this nice dendritic events in the distal dendrite. And this is also true if we do local electrical stimulation. But what about a computational model? Yes, this model can very nicely model this dendritic nonlinearity as well. This is an example, a movie, how the simulation was generated. You can see that this is the realistic morphology of the neurons, and then you can see these hot spots. This is where the a cluster of excitatory inputs are activated within the short time window, you can see locally you can generate a large depolarization and maintains the plateau potential, even measured at SOMA. So now we can do a very fun experiment, right? So using this model, we can generate this nonlinearity in the distal dendrite, and then compare it to when the proximal dendrites are activated, where only you get a regular EPSP. At the background, we can give high-frequency inputs to mimic ongoing cortical activity, and of course, with synaptic background. As I mentioned, these neurons are extremely hyperpolarized, and actually, even at 10 Hz with 10 to 20 synaptic inputs, these cortical inputs only can generate a few millivolts of inputs. But what happens now when you have the dendritic plateau potential? You can see these inputs are mimicking the 10 Hz inputs, and this is dendritic synaptic inputs. And then when they collide, you can generate a full-blown action potential. And then to do a statistical measure, we can run this kind of simulation many, many times, for example, more than 1,000 times, and get a statistical measure. So this is what happens. Without dendritic nonlinearity, you have very low percentage of generating full-blown action potential. When you do have dendritic plateau potential, you have way higher action potential generation in these neurons. And you can see the integration window is broadened. Yes?
So you just told us that, say, S1 or M1 inputs are not segregated in a particular dendritic branch.
Yes.
How do you get this plateau potential only in one segment?
So in this case, we just activate synaptic inputs within this short dendritic stretch by controlled fashion.
How would that happen in the real thing?
If you hold your question for a couple more minutes, hopefully I can answer you. So this can broaden the integration window, and also we can control the cortical inputs in the proximal, intermediate, or distal inputs. Without the dendritic plateau potential, you can imagine that the proximal inputs would have higher impact on spiking because they have fewer dendritic filtering effects. But in the presence of dendritic plateau potential, this spatial window is broadened, which means that now the neuron really doesn't care where the inputs are coming from. They are equal for generating these action potentials. So to summarize this part, how do the convergent inputs are converted into spiking output? We think that cluster synaptic inputs support the generation of dendritic plateau potential, and this can greatly broaden the spatial and the temporal window for integrating incoming inputs and then facilitate the spiking generation. And this is particularly important for spiny projection neurons because they are dominated by potassium channels on the dendrites, and they do not express, for example, IH. They have very hyperpolarized membrane potential. Previous theory work has estimated that because cortical inputs make very few connections with these neurons, we need to activate hundreds of neurons at a very high frequency to generate action potential. But using this nonlinearity, now we only require tens of neurons for generating a spiking activity. So lastly, what about inhibition? Quickly, previous work by many groups, for example, nicely summarized by Kreiser's group here, showing different inhibitor inputs in the striatum are engaging in the Phe4 inhibition, and they vary in dendritic location and also strength and the kinetics of these inhibitory inputs. Again, in the model, we can nicely insert these conductances and play with timing. In this case, we can put a PV interneuron-mediated Phe4 inhibition, primarily targeting the soma with large conductances and fast kinetics. And we can also model the dendritic targeting inhibition, for example, mediated by either medium spine neurons, or I don't know what happened, or somatostatin positive interneurons, or we can model the dendritic targeting slow kinetics iPSCs mediated by a neuroglia form of interneuron, which is MPY positive. So as I shown before, when you couple the cluster inputs and the non-cluster inputs, you can generate steady spiking output. Now you can activate the interneuron and look at the different inhibitor inputs, the impact of different inhibitor inputs on this spiking activity. First, we activate GABA inputs mediated by fast spiking interneurons. In the previous literature, it has been suggested that because they have largest conductance, it has heaviest innervation on these neurons, they should have the biggest impact of spiking generation. But however, because they are somatically targeted, which is way away from the dendritic nonlinear events, the impact actually pretty small. If we do the same simulation, now we activate these dendritic locally targeted inhibitor inputs, you can see that it has a way more dramatic effect on spiking generation. And another interesting phenomenon is that you can see this very nice window where only the inhibitor inputs coming in about 40 to 50 milliseconds delayed from the cluster inputs actually has the biggest impact. And if you slow down this kinetics even more, the NPY interneuron mediated slow inhibition has even stronger impact. So this gives us a very nice prediction. As experimentalists, we really want to test whether it's true. Actually, the timing of the dendritic localized inhibition actually matters. To do this, we went back to the experiment. I'm just showing you one of the configurations we tested. For example, we can record the spiny projection neurons and express channel-rhodopsin in the neighboring median spiny neurons. And then we know that these collateral inhibition is dendritically locally targeted. And then we can use two-photon uncaging to evoke the plateau potential, and then use either channel-rhodopsin to activate these collateral inhibition, or using GABA uncaging to activate GABA A receptors in the same dendritic location. This is what we see, that unbranched local inhibition can effectively shut down dendritic inhibition, dendritic plateau potential. But inhibition at a parasomatic location or even off-branch location has very minimal impact. For this talk, I don't have time to further go into the mechanism. It's recently published. We showed that this kind of shutdown of this dendritic plateau potential is NMDA receptor dependent. So why this matter? So this echoes really nicely with Xiaojing Wang's group's recent paper. In this paper, one group modeled the dendritic linearity in the cortical neurons. And this dendritic plateau potential is actually very similar to what we see in the spiny projection neurons. They showed that the inhibition can very nicely gate this nonlinearity if inhibition is targeting the same dendrite. With higher activation of inhibition, you can open or close this nonlinearity. And we think our mechanism is very similar, and this type of inhibition is mediated very likely by different type of inhibitor inputs. A question?
Can you go back a slide? Because there's something inconsistent between, when you pointed to that left set of traces and you said that the inhibition was effectively shutting down the NMDA.
Yes.
Typically when you shut down an NMDA regenerative event, the peak is kind of completely squashed down to nothing. It's not that it becomes narrower, it's that you would lose the regenerative spike-like event.
Yes.
And even if you look at on your next graph, when you get
like this, right?
So you're saying that the amplitudes are being depressed, so it's a different type of effect. So this is not typically seen. For example, if you put APV on, you would expect to kill that so-called spike. So you're not seeing it. I'm kind of curious what you think is going on.
So I apologize for not clear showing this data. So in this case, actually, the inhibition is delayed, is right on here. So we allow the dendritic nonlinearity to happen first, and then kill it with different timing. And then this set of experiment actually verifies what the model predicts. If we put the inhibition too early, when the dendritic plateau is generated, if we give inhibition too early, has nothing to. It cannot kill the plateau potential. You can only kill the plateau potential when you have a certain delay or you have a slow kinetics.
That's showing probability, and
it's the same for the dendritic plateau potential directly measured as well. Yeah. So, yeah, sorry, I didn't put in the detailed figure for this one. So in the end, I just want to acknowledge Yue did all the experiments and Kai did all the computational simulation. And to answer your question, yes, it indeed happens in vivo. We use two-photon imaging. I look at the dendritic events in vivo in behaving mice, and we can nicely see these dendritic events when the mice are running on the treadmill. And it has a faster onset in the dendritic location. Is way faster than somatic calcium response, which is not a backpropagating action potential mediated response. Okay, I'll stop there. Sorry for yeah, Dan.
Is the identity of individual cortical neurons that project into one of those clusters, are they functionally related? Is it a random subset? Is it a learned subset?
That's a fantastic question. So this is exactly what we're working on. We tried to trap the functional related neurons using various methods. For example, immediate early gene-mediated expression of Cre or chain rhodopsin using viral vectors, or Li Chun Luo's Fos-Cre mice. We're testing it right now. It's promising, but we didn't finish all the analysis. That's why I didn't show it. So that's our hypothesis. Actually, functionally related neurons, their projection converge on the same stretch of dendrites in the striatum. Yeah. Gotcha.
It's very interesting. I'm glad to see the awake stuff at the end, because as you know, there's good evidence that in awake animals, the spiny cells are not hyperpolarized the way that you presented them earlier. So how does that change the way we should think about dendritic potentials and plateau potentials and stuff?
Yes, so this is why we made a push, right? You see all these dendritic nonlinearities. Everything is done by either theta glass stimulation in the dendrites in the slice or glutamate caging. So we really want to see what these dendritic events mean, right? So in this case, I show here indeed it happened. And the way we want to get at, which we haven't gotten there yet, is try to look at these dendritic events and using a non-biased way to segment these dendritic activities and correlate with a behavior. For example, moving speed or paw motion in a very controlled manner. And again, this is a large data set. We haven't finished yet, but I can tell you there are way more dendritic activities even when the animals are not moving, which is more dramatic than the somatic calcium signals, which often correlate with different motions.
Maybe one more question.
Yeah.
You're showing that the main function of these dendritic nonlinearities is to drive firing.
Yes.
Right? Is there any signs that they're also inducing synaptic plasticity?
Oh yes, of course. That's another goal to understand whether these signals actually have instructive role, so they can stabilize their learning. That's where the first slide, the goal is for. We train the mice to do the food reaching task, and it's actually a learned behavior. It's not innate behavior. The performance actually improves over days. So our goal is to track the same dendrites in vivo over time and see whether dendritic activities actually plays the instruction in learning role. Well, we don't have the data yet, but that's the direction we're going. Yeah.
Great, thank you.
Thank you.